So before we talk about bone marrow responses, let's actually talk about what the bone marrow makes again, just as a review. So within the bone marrow, there's a multipotent stem cell, and this can either be stimulated to become a lymphoid stem cell or a myeloid stem cell. If I could spell, let's see, myeloid. And remember that the lymphoid stem cell, eventually, this is the common lymphoid progenitor, and then we go on to lymphopoiesis, which occurs in secondary lymphoid organs is where the maturation is, but of course there's bone marrow and thymus primarily first, and then we go into those secondary lymphoid organs such as lymph nodes, spleen, etc. And so when we talk about lymphoid diseases, we'll mostly talk about the lymph node and spleen, but realizing that the bone marrow and thymus can be involved as well. So myeloid, again, we go from that progenitor cell to a myeloid stem cell. And so the myeloid stem cell is actually going to remain within the marrow, and it's going to undergo hematopoiesis. And from here, you can get red blood cells, so erythropoiesis. You can get neutrophil production, so granulocytes, right? So granulocytes including neutrophils, eosinophils. You can monocyte production. And then the thing we haven't really talked about yet, which is platelet production. And so these are all technically myeloid cells: red cells, granulocytes, monocytes, and platelets. And when we talk about hematopoietic neoplasms, we'll talk about myeloid versus lymphoid neoplasms, and this is how we separate them. So that's myeloid and that's lymphoid. So that's one thing to keep in consideration. The location of disease, there's a lot of terms. So here myeloid means it's referring to these cell lines, but sometimes when we talk about myelogenous, we're actually talking about something that's coming from the bone marrow, and that doesn't really differentiate anything in particular. We'll also talk about something called myelophthisis, and this means that the bone marrow is being crowded out by another cell, and then we're using myeloid or myelo, but we're not referring to myeloid specifically. And I know this gets a little bit confusing. The last thing to consider is when we actually do bone marrow evaluation, we talk about a myeloid to erythroid ratio. And in this case, we're really only talking about myeloid cells and not lymphoid at all. So in this case, we're talking about myeloid really being granulocytes and the E being erythrocyte, like the erythroid progenitors. And we don't really consider monocytes, platelets, or lymphoid cells. So when we talk about bone marrow and myeloid to erythroid ratio and ME, it's really neutrophil production compared to red blood cell production. So increases in bone marrow production of a specific lineage, again, we're usually talking neutrophil lineage, myeloid, and then erythroid lineage, erythroid. So increases are termed hyperplasia, and a lack of response is termed hypoplasia. And so you cannot look at the bone marrow in the vacuum. You have to look at what's happening in the bone marrow in relation to what's happening on the CBC for real complete evaluation and being able to predict or understand what's happening in the marrow and what the response is. So an appropriate response to anemia would be erythroid hyperplasia within the marrow. And so blood loss or hemolysis of red cells would result in erythroid hyperplasia classically. And so when we have erythroid hypoplasia, of course, those are our decreased production anemias, which we talked about, and it can be specific to just erythroid or it can be generalized. And so that is when we have erythroid hypoplasia resulting in a lack of production in the periphery. Neutropenia is different, similar. So neutropenia can occur due to myeloid hypoplasia, right? So if you have myeloid hypoplasia, that can be a cause of neutropenia. However, if you have a peripheral overconsumption of neutrophils, then the appropriate marrow response is myeloid hyperplasia, and so you could see then myeloid hyperplasia. Neutrophilia is usually caused by myeloid hyperplasia, right? So unless it's mild and due to stress, any sort of inflammation is going to result in myeloid hyperplasia, which then often results in a neutrophilia. Thrombocytopenia, which we haven't talked about yet, if you have peripheral destruction of platelets, you usually then get a response called megakaryocytic hyperplasia. And Dr. Walsh is going to teach you all about that. So megakaryocytic hyperplasia. And if your megakaryocytes don't respond, then that would be decreased production and you'd get megakaryocytic hypoplasia as the cause. So those are just sort of general terminology for responses in the marrow. So of course each of the lineages can be independently impacted and you can get a decreased production or peripheral targeting of any one of these cell lineages. When all three of them are decreased, we call that pancytopenia. When just two are decreased, we call it bicytopenia, and we're going to focus on, well, you can either focus on bicytopenia or think about it, but we're really going to focus on pancytopenia in terms of the fact that when you see pancytopenia, it affects that all three lineages are impacted in the marrow, and there's either bone marrow hypoplasia, or so all three lineages are decreased in production, or there's something impacting all three lineages. And so when we talk about pancytopenia, there's a variety of causes, and these are listed in your notes as well. And so I'm just going to kind of briefly go through those. So neoplasia can result in crowding out in the marrow, causing something called myelophthisis, which I mentioned a little bit ago. And this means the marrow is crowded out and other cell lines can't be produced. And so lymphoma is one neoplasia that can do this commonly, although any sort of metastatic neoplasm can also do it. Multiple myeloma, which is a neoplasm of plasma cells, can do it. There can also be a neoplasia which eats the red blood cells, and that doesn't necessarily cause myelophthisis, it causes a targeting, and that is histiocytic sarcoma. And that can eat all cell lineages, potentially. It can also cause some crowding out, but less so. There's also infectious causes. So there's neoplasia, there's infectious causes. And we talked about parvovirus, but that was really, remember, it was limited to neutropenia because animals typically get better or they die prior to other lineages being affected. So a big one that is talked about a lot, although I can't say I've seen many cases of it, is kind of chronic Ehrlichia canis infection. Certain fungal diseases, especially if you live further south, histoplasmosis in cats, cytozoan felis in cats, and then inflammatory conditions. We have these sort of immune-mediated types of conditions that are poorly characterized. There's non-infectious causes. So a non-infectious cause that we can see is myelofibrosis, where the marrow is replaced by fibrous tissue. Necrosis, which can be idiopathic, although it's usually related to drugs. And then a relatively, I don't want to say common, but easier to identify are going to be toxic insults. And these include various drugs. So if an animal's getting chemotherapy or radiation, certainly we did the case in class of estrogen, trimethoprim sulfa can cause it. Phenylbutazone can cause an idiosyncratic reaction. Brackenfern ingestion in cattle, and even Rimadyl has been associated with it. So there's a variety of things that can cause toxic insults within the marrow and cause pancytopenia. So a history is extremely important to obtain and know what the animal's been exposed to. One thing I haven't talked about yet, but the other video is going to cover is actual hematopoietic neoplasias. And so hematopoietic neoplasias are where there is some lineage within the marrow that becomes neoplastic, and that is not necessarily lymphoma, but some sort of leukemia, either myeloid or lymphoid or even undifferentiated leukemia that can infiltrate the marrow, take over the marrow, and then can cause pancytopenias as well. So we'll talk about that in the other video for this section.